Hi, everyone. We are going through chapter 2 today. It is quite a long chapter compared to chapter 1. Chapter 1 is usually the shortest in any book, so let's get started. So first, we are going to start off very, very small, talk about atoms and molecules. We're going to work our way up, all the way up to the entire biosphere. So matter is anything that has mass and takes up space. It has 3 states, which you probably learned at junior high: solid, liquid, gas. The 4th state is a fairly new category. It's called plasma. It's kind of a thicker, gelatinous liquid. We don't go over that one too much in here. That's more of a chemistry experiment. Elements are basic building blocks of matter. They cannot be broken down by chemical means. You've all seen the periodic table of the elements. I'm going to show you again here in a few minutes. That is the official definition of it. There are 6 elements that make up the majority of living things. So in our bodies, these 6 elements make up something like 96% of us. And we abbreviate it to SHNOPS: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. Atoms are the smallest unit of an element that retains its physical and chemical properties. Atoms bond together to form molecules. Different parts of an atom, we broke that open and figured out there's different things in an atom: neutrons, protons, electrons. These should all be review for everybody. Neutrons are neutral, protons are positively charged. Neutrons and protons make up the center portion, the nucleus of an atom. And then electrons are negatively charged, and they orbit the nucleus like the planets orbit the sun in electron shells. So general structure of an atom, you got the neutrons and protons in the middle, you got the electrons orbiting around in its shell. Different elements: hydrogen, carbon, nitrogen, oxygen. They have different shells. They have a different amount of protons. They have different amount of electrons. This is how we distinguish the elements from each other. So there's your periodic table of the elements. This is kind of a fancy one to illustrate what the elements actually do, what they make up. So the periodic table, there's a lot of information you can glean from the periodic table. The atomic number is the number of protons in the atom of one element. It is also the top number in all of those little squares. So if you look at C, the carbon on the top right, you can see carbon says 4. That is its atomic number. And then there's another number right underneath it, 12.01. That we'll get to next. That is the mass number. It is the sum of the number of protons and neutrons. Protons and neutrons each have a mass equal to 1 atomic mass unit, or AMU. Electrons have a negligible mass. They are very, very tiny when compared to neutrons and protons. So they don't really have weight to add to the mass. So when we talk about the atomic mass, it's basically just how much the protons and electrons weigh, or protons and neutrons weigh, excuse me. The atomic mass is the average AMU for all of the isotopes of that atom. Isotopes. Sometimes atoms of the same element with the same atomic number, they will have different number of neutrons. So they will differ in their atomic mass. Carbon is one of them. Carbon 12, 13, and 14 are 3 naturally occurring elements. Carbon 12 has 6 protons and 6 neutrons. You add the 6 and 6, the average number is 12. Carbon 13 has an extra neutron. Carbon 14 has 2 extra neutrons. Radioisotopes are unstable isotopes. They emit energy called radiation. And this video describes radioisotopes better than I could, so I'm going to link it in a note. Molecules are atoms that are bonded together. They can be made of the same atoms, or they can be made of different atoms. And then compounds are molecules made of different atoms. Oxygen is considered a molecule. Oxygen has to bind to itself to remain stable. It can bond to other things as well, but oxygen cannot exist just by itself. It has to be bonded to something. And in the air, it's just bonded to itself. So an O2 molecule is the norm. And then water, simplest compound that we talk about. There's 2 different elements bonded together, 2 hydrogens and 1 oxygen. Electrons orbit the nucleus in paths called electron shells. Atoms are most stable when their outer electron shell, the valence shell, is full. So you have the nucleus in the middle, and then when we draw it, we draw these rings, these concentric circles going out and out and out. So the first shell is full when it has 2 electrons. The second shell is full when it has 8 electrons. The third shell is only full when it gets to 18 electrons. So the bigger the atom, the more electrons it needs to be stable. Covalent bonding happens when atoms share electrons to fill their valence shells. Each atom contributes one electron to the shared pair, and the electrons spend time in the valence shells of both atoms. Double covalent bonds share two pairs of electrons. There are also things that are triple covalently bonded. Those share three pairs of electrons. And when we draw them, we just draw them as lines. One is a single bond, two lines is a double bond, three lines is a triple bond. Ionic bonding. During an ionic reaction, atoms donate or take in electrons to fill their valence shell. Salt, NaCl, sodium chloride, is the typical example that we use. This results in the formation of positive or negative ions. Ions are just charged particles. Ionic bond is the attraction between a positive and a negative ion. So in sodium's valence shell, it has just one extra, and it's easier to just get rid of one than it is to attract seven. So it will donate because chloride has seven, so it just wants the one. So that is called an ionic bond between the two. Second section is about water. Describe the properties of water, hydrogen bonds, structure of the pH scale. So water is life. Water is the most abundant molecule in organisms, making up about 60 to 70% of our total body weight. Water is called a polar molecule because it has, just like the Earth has a North Pole and a South Pole, it has a positive pole and a negative pole. Electrons, they like the oxygen more than they like the hydrogen when they are bonded. So electrons will spend more time around the oxygen than the hydrogens, which will create a partial negative charge around that oxygen and then a partial positive charge around both of those hydrogens. Hydrogen bonds are attractions between a slightly positive hydrogen ion to a slightly negative oxygen or nitrogen. It's depicted by dotted lines, and they're relatively weak bonds. They're very easy to break. So the bonds between water molecules are hydrogen bonds. Hydrogen bonds between water molecules impart special properties. Water is liquid at room temperature. It has a very high heat capacity. It prevents large temperature changes in the body. It also has a high heat of vaporization. Hydrogen bonds must break for water to boil, and it's why sweating cools us off. A solution is formed when one substance dissolves into another, and I like to use Kool-Aid as an example for this because everybody has at least heard of Kool-Aid if they have not had it before. But in a solution, you have a solvent and a solute. Solvents take in or absorb the other substances. Water is known as the universal solvent. So you put water pretty much in any form, it's going to absorb whatever you put in it. The other parts are called solutes, and those dissolve into the solvent. So in a Kool-Aid solution, there's Kool-Aid powder that you put in. You also have to put in sugar. So there's two solutes. So solvents absorb and solutes dissolve. Polar molecules attract water, so we call them hydrophilic. Philic means to be attracted to, hydro means water. Hydrophobic is the opposite. A phobia is when you're afraid of something. So hydrophobic molecules do not like water. They're afraid of water. We also call those nonpolar. So those are synonyms for each other. Polar and hydrophilic, and then nonpolar and hydrophobic. In nonpolar covalent bonds, the electrons are shared equally. There are no partial charges like there are in water. Cooking oils are nonpolar, so they won't dissolve in water. If you've ever tried to mix oil and vinegar or even oil and water, they do not mix. Nonpolar and polar don't like each other. Water also exhibits cohesion and adhesion. Cohesion is when water molecules cling to each other through their hydrogen bonds. This is why water flows freely without separating into individual water molecules. And then adhesion is when water molecules cling to surfaces like blood vessels. More like sticking. Ice is less dense than liquid water. As liquid water cools, it becomes less dense because it expands. Those bonds spread out. So ice will float on liquid water. This acts as an insulator. Acids are substances that dissociate in water. They will release hydrogen ions into the solution. Bases are the opposite. They are substances that take up the hydrogen ions that get released, or on the other end of it, they will release the hydroxide ions. So we can split a water molecule and one hydrogen will go to one side, and then the oxygen with the other hydrogen will go to the other side. And since oxygen likes those electrons a little bit more, we call them hydroxide ions will be negative, and then the hydrogen ions will be positive. The pH scale is a measure of acidity or basicity, alkalinity of a solution, ranges from 0 to 14. 7 is neutral. The hydrogen ion concentration is equal to the hydroxide ion concentration. A pH of below 7 is an acid. The hydrogen ion concentration is greater. And then a pH of above 7 is basic or alkaline. Hydrogen ion concentration is lower. And the hydroxide ion concentration is higher. So there's just a few examples on the pH scale. You start at 0, hydrochloric acid obviously is very, very acidic. Our stomach acid is 2. Our stomach is very, very acidic, but our stomach lining is very strong that prevents it from eating away at the rest of our bodies. And then on the other side, we have pure water, 7 is neutral. On the other side, you have seawater, baking soda, ammonia, bleach. Those are all bases. A buffer is a solution that resists changes in pH when acids or bases are added to it. It is important within the body or in ecosystems as a whole. pH values need to stay within a narrow range. Just like we talked about in our last chapter, we talked about negative feedback loops. Buffers are very, very important in our body likes a certain pH. So if it strays from one side to another, it needs to be brought back to that set point. So carbonic acid and bicarbonate. Bicarbonate would be the base. Those ions act as buffers in the blood. They will absorb hydrogen ions or hydroxide ions produced by metabolism, which will prevent changes in pH and keep us at our set point. The molecules of life. There are 4 classes of organisms that are found, organic molecules, that are found within us. They are carbohydrates, lipids, proteins, and nucleic acids. The first 3 should be familiar. We eat them. They are organic molecules, contain carbon, hydrogen, and are associated with our cells, and each is composed of subunits. I know we hear the word organic and we like to think, oh, all natural at the grocery store. That's not actually what organic means in science. It just means it's made of carbon. They're carbon and hydrogens that are associated with other elements. We are organic beings. So the 2 chemical reactions that either make or break apart these macromolecules, the first one is dehydration synthesis or dehydration reactions. Dehydration means you don't have water, you're thirsty. And then synthesis actually means to combine or put together. So this is the physical act of pulling out a water molecule in order to join those 2 sugars, like in the picture. So water comes out, the 2 bigger molecules link. Dehydration synthesis. Hydrolysis is the opposite. Hydro still means water, but lysis means to split. So you are adding a water molecule in order to split those 2 into singular entities. Carbohydrates is first. These include monomers and polymers of sugar. A polymer is just a big, long chain of something, like you can see at the bottom. That is a big, huge, long chain of glucose molecules. And the monomers are the individual subunits. So each pink hexagon would be an individual subunit, would be a monomer of the big chain, which is the polymer. Carbohydrates are just sugar, just a fancy name for sugar. Most of their names end in OSE. They include small sugar molecules, like table sugar, the kind we put in our coffee every morning, or long starch molecules in spaghetti and French fries. In animals, carbohydrates are a primary source of dietary energy, and they are raw material for manufacturing other kinds of organic compounds. In plants, carbohydrates serve as building material for much of the plant body. There are simple carbohydrates. They're called monosaccharides. Prefix mono means 1, and saccharide just is the fancy word for sugar. Simple sugars that cannot be broken down by hydrolysis into smaller sugars. This is the smallest type of sugar that exists on our planet. So they're also the monomers of the longer carbohydrates. So glucose, fructose, and galactose are actually our basic three sugars, simple sugars that make up the rest. Disaccharides, again, prefix di means two, saccharide means sugar. They're constructed of two monosaccharides. They are formed by a dehydration synthesis reaction where we pull out that water molecule so the two singles can become a double. So lactose in milk is a disaccharide. Maltose in beer, malted milkshakes, and malted milk ball candy like Whoppers. And then sucrose, table sugar. These are all disaccharides. Complex carbohydrates called polysaccharides. Prefix poly means many. You should know what saccharide means by now. I've said it twice. They are made of long chains of sugar units, polymers of many, many, many monosaccharides bonded together. Starch is a familiar example of a polysaccharide. It's used by plant cells to store energy and consists of long strings of glucose monomers. Glycogen is a polysaccharide used by animal cells to store energy. It gets converted to glucose when it is needed in our cells. So, which reaction will split this molecule into smaller ones? The dehydration or the hydrolysis? Cellulose form cable-like fibrils in the walls that enclose plant cells to form wood and other plant structures. Its glucose molecules are bonded in such a way that it cannot be broken apart by most animals. We cannot digest this. In our diet, this is called fiber, and it keeps our digestive tract healthy. The next class is lipids. Lipids are hydrophobic. They do not like water. Technically, these are neither macromolecules nor polymers because they are not made of a chain of the exact same monomers repeated over and over like carbohydrates are, but they are still large molecules that give us energy. So a typical fat or triglyceride consists of one glycerol molecule joined with three fatty acids. Which reaction would join these together? What gets pulled out to link things together? Its function is energy storage, insulation, and cushioning. And it comes in two forms, fat and oils. Because triglycerides are hydrophobic, they will clump together in our body fluids. Emulsifiers are molecules that surround triglycerides and disperse or emulsify them. If you've ever washed dishes, you know, sometimes oil and fats can get on plates and pans. You use dish soap. Dish soap will cut right through that and it will dissolve the fat. That's an emulsifier. Our bile does this for us. Our bile dissolves fats in our bloodstream. So during digestion, this allows enzymes to fully break down triglycerides. Waxes is one fatty acid attached to an alcohol molecule. It prevents the loss of moisture from body surfaces. Our earwax, or officially called ceramide, is an example of a wax found in our body, a triglyceride. Fatty acids are long chains of carbons and hydrogens. The molecule always ends in a COOH, so a carbon, two oxygens, and a hydrogen. It's called a carboxyl functional group. Our bodies need a certain amount of fat to perform essential body functions like energy storage, cushions our vital organs, insulates to help maintain our body temperature. So we do need some fat. There's two structures, main structures of fatty acids. If the carbon skeleton of a fatty acid has the maximum number of hydrogens, it is called a saturated fat and its structure is straight. If the carbon skeleton of a fatty acid does not have the maximum number of hydrogens, it is called an unsaturated fat and its structure is bent because it will have a double bond there in the middle, shown in your picture. Animal fats have a high proportion of saturated fatty acids. They can easily stack, so they tend to be solid at room temperature, like butter and bacon fat. They contribute to atherosclerosis, which lipid-containing plaques will build up along the inside walls of blood vessels. Saturated fats contribute, like I said, to atherosclerosis. There's a picture. It's just plaque that builds up in your blood vessels. It will block the blood flow, which means oxygen isn't flowing properly to all parts of your body. Trans fats are typically man-made and contribute to heart disease more than any other types of fatty acid. They are unsaturated, but the missing hydrogens are on opposite ends of the carbon chain. So they're not in the middle where it's bent. Plant fats are high in unsaturated fatty acids, and because it is bent, they cannot stack to become solid because of that double bond, so they stay liquid at room temperature. Phospholipids. Its structure is similar to a triglyceride, but one fatty acid is replaced by a polar phosphate group. So they have a polar hydrophilic head and a nonpolar hydrophobic tail, which seems counterintuitive, but it works for our cells. They're the primary components of plasma membranes. They form a bilayer where the hydrophobic heads will face out the watery solutions in and around cells, and the hydrophobic tails will face inward towards each other. So you have a head, you have two tails, and then on the right is a picture where you can see how they are arranged in the lipid bilayer. There's water on the outside of the cell, there's water on the inside of the cell. So the hydrophilic heads that like the water will line both the outside and the inside, and then the tail is that yellow portion around it. Steroids are very different from fats in structure and function, but they are still considered lipids. The carbon skeleton is bent to form 4 fused rings. Steroids vary in the functional groups attached to the set of rings, and these chemical variations affect their function. Cholesterol is a key component of cell membranes and the base steroid from which your body produces other steroids like estrogen and testosterone. Proteins is our next macromolecule. They are polymers constructed from amino acid monomers. They account for more than 50% of the dry weight of most of our cells. They perform most of the tasks required for life, defense, hormones, movement, many more. They form enzymes, which are chemicals that change the rate of a chemical reaction without being changed in the process. So they're structural proteins that provide support, storage proteins, contractile proteins, transport proteins will transport substances in and out of our cells, and enzymes. Enzymes are required for our body to function properly. Amino acids are the subunits or the monomers of proteins. So all proteins are macromolecules constructed from a common set of 20 kinds of amino acids. Each amino acid has the central carbon bonded to 4 covalent partners. 3 of those partners are common to all amino acids. So all of the amino acids will have that central carbon, a carboxyl group on one side, and the amino group on the other side. It will also have a hydrogen atom on top. The 4th partner is different in all 20 amino acids. It is either called the R group or the variable group. It could be acidic, it could be basic, it could make the entire amino acid positive or negative. Hydrophobic, hydrophilic. It varies. Any chain of amino acids held together by polar covalent peptide bonds is called a polypeptide. A functional protein is one or more polypeptide chains that are twisted, folded, and coiled into a uniquely shaped molecule. Proteins consisting of one polypeptide have 3 levels of structure: primary, secondary, and tertiary. And proteins consisting of more than one polypeptide chain have a 4th level, a quaternary structure. Your body has thousands of different kinds of proteins, but they are all made of the same 20 amino acids. It's like the alphabet, the English alphabet anyway. We have 26 letters in the English alphabet, but we can make millions and millions of different words with just those 26 letters because of the way those letters are arranged, short word, long word. That is how amino acids work. Proteins differ in their arrangement, their sequence, their order, and length of amino acids, and the specific sequence of amino acids in a protein is called its primary structure. A slight change in the primary structure of a protein affects its ability to function. The substitution of one amino acid for another in hemoglobin will cause sickle cell disease, which is an inherited blood disorder. So it's very, very important that the amino acids go in a certain order to function properly. Secondary structure. The primary amino acid sequence will fold or twist in such a way to make either an alpha helix or a beta pleated sheet. Those are the only 2 secondary structures there is. And then those fold on each other to make the tertiary structure, the overall 3-dimensional shape of the polypeptide molecule. It is reinforced by interactions between the R group of different amino acids within the polypeptide. So hydrophobic are going to be folded towards the inside because it doesn't like water. Hydrophilic is going to be facing the outside because it does like water. Acids and bases are actually attracted to each other, so they're going to face each other. So Those are all different characteristics that can potentially change the way a protein folds. And then the quaternary structure forms when more than two polypeptide chains are present and bonded in a certain way. Its shape is very, very important. It typically recognizes and binds to other molecules depending on its shape and enables the proteins to carry out its specific functions within a cell. Every single protein has one specific function within our cells. It's important that those maintain the right shape, or else the function just flies out the door. A protein shape is sensitive to the surroundings. It's like us. It has a set point. It likes a certain temperature, a certain pH, and certain ionic conditions, namely salt. It likes a certain salt concentration, just like we do with our food. A negative change in any of these conditions can cause denaturation of a protein, which it unravels and loses its shape, which is what is being modeled in the GIF there. And therefore, when it loses its shape, it also loses its function. Fevers. There are high fevers above 104 in humans, can cause proteins to denature and stop functioning properly. Misfolded proteins are associated with various diseases: Alzheimer's, mad cow disease, Parkinson's disease. Nucleic acids is our last macromolecule. So DNA, RNA, and ATP are our three main nucleic acids that we are made of. Deoxyribonucleic acid is what DNA stands for. It stores information on how to copy itself. It also specifies the order of amino acids that all of our proteins need to be. Ribonucleic acid is RNA. There are various types with many functions. We'll get into them as chapters go on. Some are components of coenzymes, which help regulate enzyme action. And then adenosine triphosphate, or ATP for short, stores energy. That is our energy molecule. DNA and RNA consist of chemical units or monomers called nucleotides. Each nucleotide is made of a sugar, a phosphate, and a nitrogenous base, which is just a molecule made of nitrogen. Nucleotides are joined by covalent bonds between the sugar of one nucleotide and the phosphate of the next. That forms the sugar phosphate backbone. You can see in the picture, you have all the little P's. The little P's are connected to the pink pentagons, and P pentagon, every other. They're covalently bonded. That is the backbone of nucleic acids. There are 4 nitrogen bases. There's actually 5 when we put both together and we talk about DNA and RNA. But pyrimidines are single ring structure. Thymine, cytosine, and uracil are the 3 pyrimidines. Thymine is found in DNA. Uracil is found in RNA. So you only find 2 of the 3, depending on which molecule you're looking at. And then purines are double ring structures that just look like 2 rings stuck together, like in the picture. Adenine and guanine are those 2. So DNA and RNA always have adenine, guanine, and cytosine, but that 4th one is different depending on whether it's DNA or RNA. There are 3 basic differences when we talk about DNA and RNA. DNA, the sugar is deoxyribose. RNA, the sugar is ribose. Like we just said, A, G, C, and T are the 4 nitrogen bases found in DNA. And RNA, it's A, G, C, and U. And then the most obvious, DNA is double-stranded. RNA is only made of a single strand. Base pairing rules. They are discovered by Erwin Chargaff, so they are named after him. DNA always pairs in a complementary fashion. A always pairs with T. I like to remember it. The straight letters stick together. A and T are made of straight lines. And then C and G are the curly letters. Those always stick together. So there is always a purine pairs with a pyrimidine. It makes an exact even circle if you're looking down the barrel of it. The number of A and T molecules are always equal. And same with C and G because they always pair together. And then there's a little side note in blue there, U replaces T in an RNA molecule. Adenosine triphosphate is the chemical energy used for most cell processes. Molecules in food store chemical energy in their bond. We went through the carbohydrates, the proteins, the fats. They have very, very long structures. The more bonds they have, the more elements and molecules they have bonded together, are the more bonds that can be broken, and we can get energy from it. It's called an energy carrier because ATP transfers energy from the breakdown of food molecules to cell functions. Energy is released when a phosphate group is removed. So adenosine triphosphate becomes adenosine diphosphate. And you can see in the picture the sodium-potassium pump. Sodium gets traded for potassium. But in order to, there we go, here comes sodium. But in order to actually pump it, it needs energy. So ATP donates one of its phosphates, which gives that molecule energy to change form. So now it's facing the other side of the cell, and the potassium comes in and goes to the other side. That is chapter 2. All right, we'll see you guys next time.